little bit about protein structure, about how to predict functions based on protein structure. And I'm going to mostly be talking about analyzing HHpred data, which predicts the structure of our phage proteins and then tries to match them with known protein structures in databases of crystallized protein structures. We'll also take a very, very quick peek at a program called DeepTM-HMM, which can predict transmembrane domains as well, so that we can identify which proteins are inserting into plasma membranes. Okay, here's some review. I know in BMB 280, you have learned about the various structures of a protein. We have primary structure, which is simply the amino acid sequence of a protein molecule. And we talk about those proteins having an N terminus and a C terminus. What do we mean by that? The very first amino acid that's incorporated into a protein has one end that's not involved in a peptide bond, and that is the amine group that's hanging out on the end of it. And so we call this, because it's an amine group and it has nitrogen in it, the N terminus, okay? On the other end, the last amino acid that's been incorporated has a carboxyl group that's not involved in a peptide bond. And since that begins with a C, we call it the C terminus. So this is the primary structure of Clipper gene 8. It has an N terminus and it has a C terminus. And of course that methionine would be the first protein incorporated into, the first amino acid incorporated into the protein. This would be at the N terminus. And this valine would represent the last amino acid in a protein and the C terminus. Okay, proteins fold sort of locally using non-covalent bonding, often hydrogen bonds, to form a number of different secondary structures. One is called the alpha helix, and here it's showing all the hydrogen bonding that's occurring between adjacent amino acids that allow this sequence to form an alpha helix. And only certain protein sequences can have these types of interactions and fold into an alpha helix. Other protein sequences allow hydrogen bonding to occur between strands, such that a strand that might kind of loop around here, one part of the strand is hydrogen bonding with another strand, and that forms some kind of a sheet, and we represent these, what we call beta sheets, with these little arrows. And you can kind of follow them along. So this strand has a beta sheet formation, then there's an unstructured loop. It comes along and forms another beta sheet here, and this beta sheet is interacting with different beta sheets of the protein. So, okay. Then we have tertiary and quaternary structure. Tertiary structure is how the protein folds up into a three-dimensional shape, and that tertiary structure is often made up of unstructured terms, alpha helices, and sometimes beta sheets. This is an example of a subunit called hemoglobin. The thing is that often proteins don't act all by themselves. They actually often work with other subunits or other protein molecules. And so when they complex with other protein molecules, that is the quaternary structure. So here, hemoglobin requires 4 separate protein molecules that come together to function as hemoglobin. This is an example of a ribbon diagram of a single subunit of a terminase. And so this is like a nice example of tertiary structure where the N terminus, shown in blue, is a mixture of mostly alpha helices, but you can see some beta beta sheets, and the C terminus shown in red, with, again, mostly alpha helices, but a few beta sheets in there. This is the same information that I just provided in a different visual, where we have primary structure is simply the order of amino acids. Secondary structure is that local folding into alpha helices or beta sheets or unstructured turns. And then tertiary structure is how the entire protein chain folds up three-dimensionally. And then if that protein chain complexes with other protein molecules to form the final complex, that's the quaternary structure. All right, a great example of a protein that we know quite a bit about that has quaternary structure is the terminase. And the large subunit terminase actually has multiple domains or multiple parts with varying functions. You guys learned that a couple weeks ago. Here's an example of the large subunit terminase. It actually forms a complex in a ring in order to carry out its function. And again, each subunit, which is represented by an orange ball and a red ball, this is one protein molecule, but it has two different functions. The N terminus is shown in red, and the C terminus, the other end, is shown in blue. Here's a ribbon diagram of just that, single subunit in the terminase, and the N terminus has ATP, ACE activity, so that is one domain or one part of the protein. But the C terminus, the other domain of this protein, has nucleus activity. So again, a single protein subunit may have multiple domains or multiple parts that fold up into structures that have independent activities. All right, so we can predict the functions of proteins that share sequence similarity to proteins with known functions using the tool called BLASTP. And so here is the primary structure of a protein from the phage genome oregano, and it's almost a near perfect match to SWERTE2, terminase large subunit. 518 of the amino acids are aligning to 519 of the amino acids of SWERTE2. So proteins with similar primary structure or amino acid sequences often have similar functions, but there's something I want you to be careful about. When we use BLAST data to predict functions, we're assuming that the people that assign the function to the target protein, so in this case SWERTE2, terminase large subunit, we're assuming that those people knew what they were doing when they assigned the function terminase large subunit. So we want to be really careful when we use BLAST data to support our function assignments and make sure that we trust the original assignment in the target protein. So one way we can do that is to use other types of data which predict the structure of our proteins. So here's an example of oregano proteins. This was actually GP2, so the same sequence we're just looking at. And we've aligned it through BLAST to an enterobacteriophage, a large subunit terminase from the phage HK97. Protein structure is more like the secondary and tertiary structure is often more conserved than the primary structure. And if we look at this alignment, we can see that this is a really poor alignment. These two sequences don't share a lot of sequence identity. But they do fold up into similar tertiary structures and probably have the same structure, the same function rather. So how do we learn what the tertiary structure is of oregano GP2? Let me first refer you back to, this is the structure of the HK97 large subunit terminase that we looked at before. It has this particular three-dimensional structure, and somebody has determined this what's called the crystal structure. They know exactly how this protein folds, and they know where every single amino acid is in the three-dimensional structure down to the angstrom. What HHpred does is it predicts the structure of the oregano sequence based on its primary sequence. So based on the chemistry of the amino acids and the order in which they occur in the primary sequence, they can predict what both the secondary and tertiary structure is. Let me show you how to read this alignment. So very quickly, on the top is the sequence of the oregano sequence, or a query protein. And on the bottom of the alignment, right here, is the sequence of whatever the target protein, the hit the HHpred found. And in this case, it is our friend, the terminase large subunit from HK97. Below the sequence of the target protein is what the known secondary structure is. So red H's represent alpha helices, and blue E's represent beta sheets. So for the HK97 protein, we see an alpha helix, an unstructured coil, a beta sheet, a coil, and then another alpha helix. Above the sequence of our query protein, oregano 2, we see the predicted secondary structure. And it also has an alpha helix followed by a coil, by a beta sheet, a coil, and another alpha helix. And if we follow along all the different rows of this alignment, that we see for the most part that they have the same secondary structure. Let's look at this even more closely. So in between the two sequences is what we call the consensus, and there's all kinds of markings in here to indicate where the sequences either have identical amino acids, so here's a glutamine that's found in the same position in both alignments, or there's indications that there's similar amino acids. So each of the amino acids has a particular color, and that is also meaningful. So for example, amino acids that have the same color have the same chemical properties. Red means either arginine or lysine. These two amino acids have positively charged R groups, whereas these blue residues, or amino acids D and E, represent aspartate and glutamate, and these have negatively charged R groups. They have carboxyl groups hanging off of it. The green represents hydrophobic amino acids. And so where they may have different amino acids there, whether it's an isoleucine or a cysteine, they have similar properties. Okay. Other things we want you to look at are the probability score. The probability score tells us the probability that our match is high quality and not just random. And we will only look at HHpred hits that have probabilities of 90% or above. You can also see the E value, which of course is the probability that the match is random rather than specific. So again, we want a really, a number that approaches 0. But we will look at E value a little bit less heavily than we do with blast data, because the E value is going to be impacted by the length of the alignment. What we care most about is actually probability. Okay, next, the other thing I want you to look at is which amino acids of our query protein align with the terminase protein. So here we can see the 59th amino acid of our query protein aligns to amino acid 36 of the target protein, and the alignment goes through 493 of our query to 499 of the target. And the number in the parentheses tells us the total number of amino acids in each protein. So this gives us a sense that that our protein is aligning to most of the amino acids of the target protein. So this will be really important data to keep track of, is the total coverage of the alignment. The other thing that we can look at are motifs in the alignment. So I've shaded, I went and had to read a paper, and what I found out from the paper was where in this alignment of the HK97 terminase, what part of the protein is involved in the ATPase domain. So again, I looked this up in a research article and found that the amino acids that I've highlighted in yellow are part of the ATPase domain, and I can see that the alignment includes the entire ATPase domain of the HK97 protein. The other thing I did was I looked some things up and I asked myself, what kinds of motifs or amino acids have to be conserved in the sequence in order to function like an ATPase? And again, these are things I looked up. I learned that there needs to be a Walker A motif and a Walker B motif in order to be an ATPase, and I found these sequences are conserved in the sequence. Okay. And then the C terminus, again, I looked up to see which amino acids in the terminase are involved in the nuclease domain, and I have highlighted those. Essentially, what I've shown is that this alignment includes both the ATPase domain and the nuclease domain, and so I feel pretty confident in calling oregano 2 a large subunit terminase. The last thing I wanted to show you guys was that sometimes we can't come up with, we don't have good data from HHpred to give a really specific function. Another thing that we can do to get at what the function of a protein might be is to determine whether it's globular, and globular means it's just part of the cell's cytoplasm and that it's soluble, or is it inserted into a membrane? And so we use programs to detect alpha helices that are inserted into the membrane. So here's an example of a protein that has a single transmembrane domain, meaning it goes across the entire membrane. Here's a protein that has 3 transmembrane domains, and it's almost like a sewing needle just sewed the protein in and out. There's pieces of the protein that hang out in the cytosol, and there's pieces of the protein that are hanging out in the extracellular environment. Here's a protein that has at least 5 or more membrane domains. So let's look at what that looks like in Pecan. We use the program DeepTMHMM for transmembrane prediction. Here's a protein that is globular, and this little graphic is showing that there are no transmembrane domains. The x-axis shows the sequence down below, and the y-axis shows the probability that parts of that sequence are involved in a transmembrane domain. Here's an example of a protein from Clipper that has 4 transmembrane domains, and again, this is giving us an indication of which amino acids along the sequence. It looks like it's a little over 140 amino acids. Which of those amino acids are involved in the membrane domains. That's all for now, and we'll learn more about how to interpret HHpred data in our Friday seminar in a couple weeks.